I think you unenabled the waiting room too, right, Prabhakar? Prabhakar?
I just did.
Okay, so people can come in. All right, welcome everyone. This is Dr. Shiva. I also have my colleague, Dr. Prabhakar, with us, and we have a couple of other people from CytoSolve with us, including, I think Ken Fielding's also here. Ken, are you here? So let's share what we're going to do. I'll do an overview. We have decided to do monthly symposiums on CytoSolve. For nearly 16 years, we've worked on building out the CytoSolve technology platform, the business process, and a very powerful capability that's really the future of medicine. And that capability involves a groundbreaking technology framework that we've been working on for nearly 16 years. What I want to share with you is the fact that this technology framework can be used in any field of science or human interest where it involves biological sciences, life, and then we can use it to identify things that we consume, things in our environment, not only for us, but animals, plants, humans, all life, the interaction of those to figure out what supports and advances life and what destroys life. And this is at the molecular systems level. We shared, Prabhakar, when did we do our last symposium? Last month, right?
Yeah, 2nd of April.
2nd of April. So we've decided the second Tuesday of every month, we'll do a symposium on a topic. The first symposium we did was on animal testing and how CytoSolve ends the need for that. And we discussed how there's no need for animal testing, independent of us and the technology we have. Pretty much most people have looked at this, realize that animal testing is unnecessary. But today we're going to go a little bit more into the platform and going to give you an example of the fact that every major human disease or biological function, CytoSolve can tackle. And when I mean tackle, which means step one, we can build a deep scientific understanding at the molecular systems level of that disease or biological phenomenon. So you may want to take a note on that, number one. And we visually and scientifically represent that as a systems architecture. That's step one. So this entire paradigm is what I created in terms of going end to end to revolutionize medicine. So one is we have a systems architecture of a disease or a phenomenon. The second is we have a very uphill task because what we're doing is revolutionary and radical, but yet we have to get quote unquote credibility on it. So we have to publish these things in peer-reviewed journals. So the second part is publication. And we figured out how to get leading people in the field who are also revolutionary in their own world, who typically people who have tenure in some sense, but don't care anymore about serving the establishment. So they have become partners in helping us get out to the rest of the world that this stuff really works. So once we do an architecture of a disease, we then publish it. Once it's published, we convert the individual diagrammatic molecular pathways using mathematics to the quantitative predictive version of that pathway. So every piece of what we create, it's not really a model. We're literally, it's backed up by the individual science experiments. So we aggregate all this, have figured out CytoSolve is a very powerful technology that allows us to scalably integrate this and then compute it. And then once we've done that, we then have the ability to use this quantitative understanding of a disease, predictive and quantitative understanding, to then test using the computer without killing animals whether how individual interventions affected or combinations of interventions. It's like a combination of compounds. And that leads us to figure out what works, what doesn't. And then in order to get protection for the discoveries we make, we figured out how to do patent protection. And then from that, we can then use that to start creating new products, new research, new publications, new discoveries, which we then make available to the public. This entire process over the last 16 years, it's a long haul. We now, as we'll go through this towards the end, we want to make the entire process and platform accessible to all of you. While we have done some heroic efforts here and some revolutionary work with this development, in order to get this out there, we're going to need you guys, meaning each one of you on this call can yourselves become a citizen scientist, an inventor, a discoverer using our technology framework, because we can do the research faster, cheaper, and better than, if not all or most of the large universities, and we've proven that. So think about what we've created. We've created a powerful technology platform without the need for animal testing, which can find compounds in nature that work to create new products. And imagine each one of you doing that. So we're going to walk you through today is we're going to take, so we're right now going after 35 of the major quote unquote diseases in the world. And I put diseases because ultimately these diseases are not really diseases. They're basically dysfunctions in different molecular pathways. Western medicine calls them disease, but traditional medicine says all disease has one cause and one solution, which is, nutrition ultimately. Okay. What you eat, what you do, your lifestyle. So we're going to today take on something called pain, joint pain, inflammation, osteoarthritis, and we're going to walk you through what is the solutions a swarm offers today, what the solution that the big vitamin guys offer today, which also are ineffective, and then using this very powerful technology in a very profound way, how we've aggregated all the research out there, identified the individual molecular mechanisms, which are little pieces of truth, connected them together, converted them to these quantitative models, and then figured out a very powerful combination of ingredients which have profound effect for alleviating. We have to be careful. The FDA, we cannot say the word pain. But we're going to say alleviating natural discomfort. And that's what we're going to talk about. And that's a product called MV25. But this is not a product advertisement seminar. This is more to show you that we fully vertically integrated, from doing the basic research to publishing, to doing the modeling, to doing the screening, to getting the patent, to manufacturing. And all those steps, all of you can also do if you seek to do it, or you can just support us in supporting our research. We'll talk about that. So let's begin. If Prabhakar goes to the next slide. So those of you who are new, my name is Dr.SHIVA Ayyadurai, and I've been at this for a very, very long time. I'm an MIT PhD, a working scientist, and many of you know I'm also running for president of the United States because it's our view that the people of America really need people who actually know how to solve problems. CytoSolve is one of those solutions. Next. So Prabhakar, maybe you can go through what we're going to learn today.
Today we'll start the presentation with with understanding the power of system science and how it connects not just science but also politics, health as well. And then we'll go over what is CytoSolve, and then we'll go into the joint health mechanisms behind it, molecular mechanisms, and what CytoSolve has been able to do in that field. Then we'll also go over how MV25 was developed using CytoSolve. then how CytoSolve is able to do the research faster, cheaper, and safer. Then we'll end with how you can support our research.
Great. So let's go to the next slide, Prabhakar. So let me jump back in here. Thank you, Prabhakar. So I want to play a video that'll hopefully inspire you to let you know that each one of us has a journey, but everything we do is we take a systems approach. And this video will share with you my journey to take the systems approach, which imbues everything we do: CytoSolve, Systems Health, Truth Freedom Health, Clean Food Certified, all the solutions we have. Running for political office, we take a systems approach to everything to identify the real problem and the real solution. You make sure the volume is on, Prabhakar, so they can listen to it. Yep, go ahead.
Welcome to VA Shiva. VA Shiva is a product of my journey across East and West, science and tradition, ancient and modern, that brings you the science of systems so you can become a force for truth, freedom, health. VA Shiva is a platform of revolutionary education, community building, and weaponry for unleashing local activism. My journey to VA Shiva begins in the chaos of Bombay, where I experienced diverse religions, languages, castes, and in a small village that had no running water, no electricity, where my grandmother, a poor village farmer, practiced Siddha, an ancient system of Indian medicine over 10,000 years old. She observed one's face, the art of Samudrika Lakshanam, to understand a body's unique constitution, allowing her to deliver the right medicine for the right person at the right time. Watching my grandmother heal others, I was inspired to study medicine, but I was also aware of the corrupt caste system of India, which denigrated a human being, where my family were considered low caste untouchables, where one's birth determined one's destiny. The grit and determination of my mother and father led them to get educated and to come to America, a 1 in a trillion event. Their actions inspired me to work hard and excel. While in 9th grade, I attended New York University in a computer science program, and subsequently, at the age of 14, was given a full-time job as a research fellow at what is now known as Rutgers Medical School in the heart of Newark, New Jersey. There I investigated the system of sleep using mathematics, computer science, and biology for sudden infant death syndrome. It was there in Newark where I invented email, when I was the first to convert every feature of the physical, paper-based interoffice mail system, including inbox, outbox, memo, carbon copy, blind carbon copy, attachments, into its electronic equivalent, a system which I named email, a term that I was the first to coin. On August 30th, 1982, I was awarded the first U.S. copyright for the invention of this system, recognizing me as the inventor of email. At that time, copyright was the only way to protect software inventions. I went on to MIT, where I earned 4 degrees across multiple systems of engineering: electrical, mechanical, design, biological. That training led me to invent many other systems for advancing humankind. EchoMail, the world's first intelligent email management system. CytoSolve, a computational biology system for eliminating animal testing to discover new medicines faster, cheaper, and safer. And to the creation of a whole systems methodology for certifying clean food. These innovations led me back on a Fulbright to India, where I discovered the missing link between Eastern medicine and Western systems theory, honoring my grandmother, to develop Your Body, Your System, a powerful tool that will help you understand how your body is a system and how the inputs of food, supplements, and exercise bring your body back to its natural system state. And Systems Health, an integrative educational discipline that is now integrated into the VA Shiva platform that will enable you to learn the science of systems, the science of everything, be it your body as a system or our society and politics as a system, to reveal the foundational interrelationships between truth, freedom, health. Now is a time for you to be the light, learn the science of systems, build community, and weaponize yourself to unleash the activism necessary to deliver truth, freedom, health in your local community. Welcome to VA Shiva.
All right, thank you, Prabhakar. Let's continue. So welcome, everyone. This is Dr. Shiva Ayyadurai. If you made it, you are at the second CytoSolve Symposium we're doing. And today we're going to be discussing joint health, the science of it, the molecular science of it, and CytoSolve, and a very powerful solution that came out of this platform, and then how you can all be part of it. Next. So as the video shared, we take a three-part integrative approach to everything we do. We have to think, fight, and heal. Next. And to support that, we have three major solutions. Truth from Health is really the Leadership Institute. Next slide, Prabhakar, if you can sort of continue. Which it really educates all of you to really become leaders by understanding the science of system. We've made it very accessible, affordable to anyone on this planet. So if you're not a Truth from Health warrior, if you are, or you were one, become part of that, because it's a foundational element. The other piece we have is a Systems Health Institute, which really is training people over the last nearly 15 years, 14 years, on how to become a health educator. There's a world of coaching that's developing for health coaching, but it's very sort of a namby-pamby curricula others have developed. We've developed a formidable curriculum that'll help you really understand the body as a system and educate others. You can use it for your own health, for others, but it'll help you navigate through the myriad of solutions to figure out what's the right solution for the right person at the right time. So that's a very powerful platform too. CytoSolve is what we're going to be talking about today. And CytoSolve is really part of our Open Science Institute. It really powers it, so all of you can be part of that. Next. So just quick announcement, Truth from Health meetings we do every Tuesdays at 11 a.m. and 8 p.m. You can go to truthfromhealth.com. Come to it if you've been sort of hanging out. I know a number of you probably got involved when I exposed all the stuff with the pandemic and the election stuff, but we want to inspire you and educate you guys to get off your butt and get involved, because the world is not going to change by being entertained. It's going to be changed by you guys getting involved. We have to get on the ballot in about 50 states in the United States to become part of that. Wherever you are in the world, we have many, many different things you can do. So come to the open house at 11 a.m. or 8 p.m. and you can go sign up for the orientation. Next. The Systems Health platform, just quickly, we have a major summit coming up this May, 24th to the 26th. Prabhakar, you can probably share them the link. Prabhakar will put the link up, but we want to build an army of systems health educators all over the world. You can use this knowledge to start your own educational business, advise other people, etc. from an educational standpoint. I have a black screen, Prabhakar. Something happened?
I'm trying to send the link.
Oh, okay. I see when you send it. Okay. Yeah, so Prabhakar will send you the link, but this May, we're having a summit, which is one part of the larger certification and online program. Most of it's done online. If you can't physically come to Cambridge, you can also do it online. All right. Thank you, Prabhakar.
All right.
So the other piece is that we do tutorials on this every other Fridays on 11 a.m. and 8 p.m. as Prabhakar sharing here. And if you want to go to those tutorials, you're welcome. Next, we also have CytoSolve, which is what we're going to do today. And we're having our symposium. All right. I think it's second Tuesdays, right, Prabhakar?
It's the first.
Oh, is today the first Tuesday?
Yeah.
Oh, okay. All right. So let's talk about molecular science of joint health next. So let's first of all let you know, our goal at CytoSolve is to really revolutionize medicine. We're the future of medicine. And what we're doing is figuring out what works at the molecular level, as I mentioned, and this could be if you're building functional foods, if you're building nutraceuticals, therapeutics, what combinations work right for the right person at the right time. And that's really the revolution. Right now, it's all guesswork. It's all sort of, so pharmaceutical companies stay in the world of, the world of synthetic compounds that they can control, but it has a lot of side effects. The big vitamin guys are throwing a bunch of crap in, but if you ask them, why did you throw this stuff in? They can't really explain. So a lot of the stuff is it is indeed snake oil and doesn't have efficacy. So our stuff helps figure out what works and what doesn't. Next. So this is the way that traditional pharma companies, conventional, do research. The big vitamin companies don't even bother doing this. So one could argue they're bad or worse. They throw stuff together. Pharma can only handle a single chemical compound. They do a lot of test tube testing in vitro. Then they kill a bunch of animals. That takes 6 years. And then they get approval from the FDA to go do animal testing. The problem is, as you notice in the first bullet, less than 20% entering phase 1 of clinical testing are even successful. And it costs them a lot of money, a lot of time. And then most of the products that they do create side effects. You can see with this new drug they're putting out, their Ozempic, it has people take it, they get all sorts of side effects, but it's not addressing the underlying issue, which is the fact that food systems in the United States have been completely destroyed. And it doesn't educate people on how to eat properly, processed foods, et cetera. So this is essentially a reactionary model, and it's very consuming, and it's a medieval model. Next. And this model is failing. Pharmaceutical companies, as you noted in the orange line, are spending more and more money on R&D, as Prabhakar sharing that tracing of that orange line, and they're getting less and less new discoveries. So it's a failing model. This is why they needed the vaccine and the mRNA platform, because this gives them a new way of getting out of this. And this is why Trump was invaluable to big pharma. Next. You'll notice that the reason this failure is taking place, so we're going to talk about osteoarthritis today. As we'll walk you through, osteoarthritis involves hundreds of thousands of chemical pathways. So if osteoarthritis is represented by the elephant, the individual researchers are not incentivized to put all that together and build a systems model, which is what we do. The individual researchers, like blind men, touching pieces of that puzzle. Next. And because they live in this isolation, if they ever put it together, they wouldn't get anything that looked like the elephant. They'd get something like this. Next. So the traditional model of development is really sort of trial and error. They come up, it would be like we came up with an old, in the old days, an airplane design. We threw the pilot in. If it crashed, you said, gee whiz, it crashed. And then if something succeeded, you acted smart and you try to back explain why. But there's really no fundamental laws that are helping development in biology. Next. So as I shared, there's a need for a revolution. And in the interest of time, as I shared with you in that video, the revolution here for me began when I was a kid doing research as a 14-year-old on why babies were dying in their sleep. Again, the common theme was it was a biological system. The dysfunction was the babies dying in their sleep through what's called an apnea. So I used mathematics and physics and science to look at their sleep patterns and try to correlate that sleep patterns to the baby's death. And we made some very, very interesting headway. But while I was working at that hospital, not only did I develop a love of computing and biology, but also, as Prabhakar will walk you through these slides, as I shared, large-scale systems. Email is really a system. It's not the simple exchange of text messages. CytoSolve, as you'll see, is a system. Here, email is the interconnection of inbox, outbox, folders, hundreds of different components connected together to enable us to do actual email, not simple text messages. I wrote the code, got the first US copyright. All of this occurred, by the way, before I came to MIT, not in the military industrial complex, military, but email was created in a medical school, solving a civilian problem. And all the proofs and all the receipts are there. There's no controversy except the one that frankly, the racist pro-military people created, because they cannot have innovations coming from the edges. It has to be from their centers. So anyway, but I learned how to create large-scale systems next. So when I looked at the problem of taking place in medicine in 2003, when I headed back to MIT, this graph represents the fact starting from the left to right. When the Human Genome Project started, we thought we had about 100,000 genes. We knew a worm had only 20,000, so we thought complexity was a function of the number of genes. Well, what you see in this diagram is, as the right, we actually have the same number of genes as a worm. We are not genetically, in terms of the number of genes, we're in fact about the same. Corn and plants actually have more genes. So this led to a field called systems biology. And systems biology basically said, if we're going to understand the whole human body, it's not just the genes, it's the proteins, the molecular reactions, the cells. So you have to integrate all this stuff. Next, and at that time, in 2003, when I came back to MIT, the reason I came back was the United States government and the National Science Foundation had said, okay, let's move out of the DNA and let's start looking, or the nucleus, and let's start looking at the entire cell, which is much more complicated. And the notion was, could you mathematically model the whole cell? Because if we could do this, we could frankly eliminate completely the need for animal testing. We could do predictive biology. So that's what I came back. And over the next 4 years at MIT, 3 years, I'm sorry, 4 years, 2003 to 2007, I created a technology called CytoSolve. It was the beginning of it. So the idea was, if the body has all these molecular pathways, which are chemical reactions, could we convert that to models? And that was happening. Next slide. And then could we interconnect all these systems of models? If we could do that, we could predict biological phenomenon. This was seen as an impossible problem. I took it on, and the approach I took was to treat the cell not as just one big lump, but a system of interconnected molecular pathways which we could model. Next slide. And CytoSolve would be this technology which could integrate all of these on the fly. And that's what we created. Next slide. It took me a lot of years to get this published because it was seen as very revolutionary. This paper took 3 years to get published. Next. And then the next part of that was recognizing if this is a traditional drug development model, what's next slide. And what CytoSolve is a much more radical model, is a feedback system. We can handle multiple compounds. If you look at products in nature, if you eat an orange, if you eat, a ginger root, if you eat, whatever, anything, spinach, whatever you're eating, they're not just made of one chemical compound, they're made of hundreds of compounds. So if you want to really understand how food works, you better be able to handle not just a single compound. CytoSolve allows us to do that without the need for animal testing. Next. So this is how we build airplanes. So this is the revolutionary nature of this. And as I mentioned, this platform is now available to all of you if you seek to use it. Today, we're going to talk about, out of the 35 diseases we're going after, how we're looking at osteoarthritis. So anyway, we have a global collaboratory in our data center. We, every day, we're taking new areas. We're looking at the publications. We bring in different people from the research community, and this is an iterative process. But that is CytoSolve. Next. And using this, we have a very methodical process. First, we can look at all the literature. Step one, we can then mine the literature. We can extract out the chemical pathways. Then we do the modeling, and then we do screening. Next. So that's what we've created. And we've worked with those who want to help us. We thought pharma would want to use this to create things that didn't kill people. They would start using us and realize that we would potentially find things that were not glamorous for them, and they'd exit. Universities. We work with the largest universities in the world, Stanford, UCLA, Harvard, MIT, who come to us because we can do the research faster and cheaper. So note that. This is why I say it's revolutionary. We work with government, the NSF, NIH. We've done lectures for them, foundations, right? Anyone who wants to listen, because in many of these even large organizations are some radicals. So over the last 16 years, we've proven this technology works. We've published many, many papers. Next. And people have used us in a variety of different ways. You can look at, people have used this technology for all different things that we never even thought possible. Next. But my interest is really in natural compounds. Let me just walk you through an example. So here's an example of the University of Southern California outsourcing its research to us and how it led to a major publication in one of the most prestigious journals in the world, Nature Neuroscience. So when you look at the body, if you look at your brain, next slide, you'll notice that your brain is covered by a lot of blood vessels. And we're looking at one of them. In fact, the blood vessels would go from San Francisco to Los Angeles. That's how many blood vessels you have. The cross-sectional view of that blood vessel is composed of the endothelium, as Dr. Prabhakar is pointing out, which is a cell. And then it's covered by another cell called the pericytes, which communicates with another cell called the astrocytes. So 3 cellular components. When the pericytes, it's not parasites, break down, you have all sorts of disease. So next slide. So what we did was we used CytoSolve to map out every molecular pathway in the endothelial, next, every molecular pathway in the pericytes, every molecular pathway in the astrocytes. And by the way, every ball and stick diagram, we can show the exact experiment from which it came from, the actual scientific truth, little pieces of truth. Next. And then here, the major contribution, which was really that I made to this paper, was organizing all of this from a systems architecture. It's like if you're a blueprint for a house. And biologists really weren't even looking at the world this way before I came along, this paper came along, which is on the bottom layer, you have the endothelial, pericytes, and astrocytes. The next layer, you have the functional messaging between the endothelium and the parasites, same with the parasites and the astrocytes. And then we layered in the disease. So what you can see here is disease is not, frankly, a disease. Disease is a particular dysfunction in that particular pathway. So if you look at ALS, which is the Lou Gehrig's disease, up on the middle right, or Alzheimer's disease, or you notice they're the same dysfunctions in a particular pathway. So the way we as engineers look at diseases, it's at the molecular pathway level. So there's really no reason to call something a disease, in a sense. It's a label for particular dysfunctions, but they're all interrelated. Okay. So this was published in Nature Neuroscience. When we first submitted it, the reviewers thought it was brilliant. Half of them, and the other half of them thought I was a crackpot to even use these words, systems architecture. Anyway, it got published. It's got thousands of citations. To give you an idea, there are people who publish paper and they're lucky if they get 1 citation or 5. This is 1,000. So it's become a landmark paper in the field. Next. The next thing is we can use CytoSolve to do very powerful kinds of predictive modeling. Then people say, well, does your models match reality? So Prabhakar, if you can walk them through this slowly, I'd appreciate it.
So this is a project that was done in collaboration with 4 different institutions, including MIT, Harvard Medical School, and King's College London, where the researchers, they were looking at vasodilation, or the process by which nitric oxide is produced. When you exercise or when your blood is pumping, it passes over a thin layer of cells called endothelial cells, and this it exerts a shear stress on the surface of the endothelial cells, and that results into formation of nitric oxide through L-arginine, a reaction that is catalyzed by an enzyme called endothelial nitric oxide synthase. So it's a very critical process to life. It maintains vasodilation. At the same time, when you look at the cellular surface and what exactly is going on, what you see, it's an expanded view of the endothelial cells here. There is a structure called glycocalyx on the surface of the endothelial cell. When blood flows over it, it actually mechanically shakes, and then that results into production or activation of the endothelial nitric oxide synthase, which then catalyzes the arginine into nitric oxide, which further down the line relaxes the smooth muscle cells. So if you look in the literature, there are these various explanations of what is going on at the cellular level, but there's no coherent view of what's going on. So the researchers there were trying to understand how exactly shear stress is responsible for producing nitric oxide, and what are the molecular mechanisms. So we came in, so we used CytoSolve to individually model these independent pathways, then combine them to understand the system of production by modeling these individual pathways and combining them with the CytoSolve platform. So what we see here, these are the results from our in silico analysis or experiments. So this, the parameter that we are measuring here is called the eNOS mRNA, which is the mRNA that produces the protein. And we plotted it against the time. Once we had that, we actually overlaid the experimental data that researchers at Brigham and Young were doing. And as you can see, these are some really excellent results. They overlap with our in silico predictions very well, indicating that the CytoSolve mathematical model is actually replicating the reality inside the blood vessel. So similarly, for another variable called enos protein, this is the CytoSolve's results, and then overlaid with the experiment data, which also matched pretty well. These results were published in Cell's Biophysical Journal, a pretty highly respected journal from the Cell Press, and Dr.SHIVA is one of the co-authors there. So what this project shows is that we not only can create systems architecture like the neurovascular diseases, but also can develop models based on that systems architecture, and then combine those individual models to predict a biological phenomena like the production of nitric oxide using these models. So people thought that it's kind of an impossible task, but CytoSolve was able to predict as close to reality as possible using this novel approach. So the next case study we have is another collaboration with a company, pharmaceutical company called Alnylam. So they're a pretty big company that specializes in siRNA. So the founder of that company actually won a Nobel Prize for discovering the silencing RNAs. So they were looking at a disease at that time called, it's a rare disease, affects the liver, called hereditary angioedema. And they had a lot of preclinical data, a lot of animal data, but they were kind of hesitant to move forward into the clinical phases because they were not really sure of what is the mechanism by which some of their therapeutics were working. So they came to us and asked us if you can model it and predict whether predict the actual mechanisms of action for this particular disease. So we spent almost a couple of months to understand the system, created the systems architecture. For the reasons of confidentiality, I can only show you the larger aspects of it. So there we've identified two systems, the contact activation and bradykinin, that govern this particular disease. And we modeled these two systems and then created these in silico predictions. So on the left side, we have a, so this is one target that they were targeting called FX2 or F12. And they were trying to use the siRNAs to suppress this. And on the left side are the CytoSolve results. So we're able to predict the amount of biomarker of bradykinin by the suppression of F12. And on the right-hand side, we have the mouse data that we got from the client. And as you can see, it matched very well with our prediction, confirming that this is a right target for their therapeutics. Similarly, another target, high molecular weight kynurenine. Same here, this is the CytoSolve data. And then when we finished that, they gave us the mouse data, which also matched pretty well with our predictions. So they were so impressed with our results that they actually gave us a very excellent press release saying that what we have done actually helped them with their clinical decision. And they actually were able to accelerate the drug development process based on our data.
Yeah, so I think the main goal between these two examples is we have shown, with the three publications and commercially, with these, very, well-known biotech companies that we can, even though we were only, I think, 3-4-person, 5-person company at the time, that we can do research at a level of a multi-billion-dollar company. And that's a key takeaway. Because people say, oh, does your stuff work? Does your stuff work? Prove it to me. Prove it to me. That's important. The next one, Prabhakar, is even more compelling. Yeah. So what's compelling about this, again, we want to give you guys enough data points that the establishment themselves has recognized the power of this. But the problem is the establishment has no interest, in our view, to do things faster and cheaper. They claim they do. So this is an example where this paper came out, again, in one of the #1 journals in the world, Nature, saying that if you're going to solve cancer, you have to use combinations of compounds, which is what food is, okay? But in the case of cancer therapeutics, they use chemical compounds, right? Synthetic compounds in combination. The thing is, but how much, if you have, let's say you want to do 4 or 5 combinations of cocktail, the doctors have no idea how much to use. How much is very important. One dosage can heal you, the other dosage can kill you. So the authors of this paper, who I do not know, so there's no inside job, had referenced my PhD work as the only capability that could really go at this. So when this came out, we raised a little bit of money. First time I took in any investment money from people who actually were interested in natural products. And our goal was, could we, in under 12 months, look at those compounds that already existed, combine them into two compound combinations, and discover something that could do better than the existing drug for pancreatic cancer. So we went down this process and we modeled all the molecular pathways of cell death and cell proliferation, which is metastasis. We ran through trillions of combinations. We found that 70 were viable, 5 we optimized, and 1 we applied. Again, we were doing this as an exercise. At that point, we're, I think, a 7-person company in Cambridge. And we wanted to see, without killing animals, could we submit the data and get it allowed by the FDA? And after we submitted this, we got a call from the FDA about 30 days into this, and they said, we're very impressed by what you guys have done. This is the future of 23rd century medicine. And then they gave us the allowance. Now, we didn't know what to do with at that point, so we went to MD Anderson, who's one of the big cancer centers, and we wanted to see if they were wanting to do anything with it. And then we created a partnership where they also wanted to integrate natural compounds. But the point is, again, we've played with the big guys. So no one can say, oh, your stuff doesn't work, show it to me. Well, we can show you many, many examples of where this platform can work. So this is yet another example. Now, let's move on. And by the way, because my love is really natural compounds, if you look at in traditional systems of medicine, when people combine stuff, they'll take a little bit of this, a little bit of that, put it in mortar and pestle. And if you ask them, how did you come up with this? The healer can't really explain. But with CytoSolve, by way of looking at inflammation, you can look at all the ingredients in, or the compounds in curcumin, the curcuminoids, and we've mapped out all the chemical reactions going through 6,000 papers where they hit. And we looked at all the papers where resveratrol hits. So again, this is the example of having your curry meal, you're eating an Indian meal with red wine. And then what happens if you want to figure out how much curcumin and how much resveratrol you need to take? Here, through CytoSolve, we're able to do in silico, which means on the computer experiments. It's the first case we don't give any curcumin, any resveratrol, and we're simulating high levels of inflammation, which is represented by that number 0.15. Then we give a little bit of curcumin, but no resveratrol, and you notice it lowers the inflammation. Then we give a little bit of resveratrol and no curcumin, and then that lowers it, not as good. But then we lower the levels of curcumin even further and lower the levels of resveratrol even further. And you notice it has a tripling effect, because this is called a synergistic effect. And this is why food is medicine. Okay. So anyway, that's the background on CytoSolve and how all of this works. Again, all of this platform is, we've now, independent of us using it to discover new things and help other people, you can also start using this platform in a model we'll talk about. Let's go to now joint health. Prabhakar, can you walk people through slowly joint health, what it is, and then walk them through the UHN work we did? Thanks.
All right. So just a kind of an overview on joint health, and especially its symptom, joint pain. It's been known that not just the US, but worldwide, about 500 million people are being affected by the joint pain, and it is caused by several diseases such as gout, fibromyalgia, lupus, rheumatoid arthritis, and osteoarthritis. And it affects mainly the joints, all the joints, and then it basically degenerates. And as the joints are degenerating, there is a sensation of pain that gets worse and worse. What are the treatments that are currently available? So one is the surgical treatment where they replace the joints, but most of the people actually get the non-surgical treatment. So this is kind of a graph of what type of treatments are there and what percentage of people are actually taking those types of treatments. So the most common treatment these over-the-counter NSAIDs like ibuprofen and Tylenol. 66% of those patients use that. Then we have supplements. 26% of people use that. Steroid injections for people suffering even more pain. There are also non-opioid prescription pain medications and opioid prescriptions as well as oral steroids. Most of you probably know that the NSAIDs like ibuprofen and Tylenol have severe side effects. Here's a list of it, but briefly, you can have side effects such as heart attack, stroke, high blood pressure, kidney problems, digestion, gut problems, stomach pain, constipation, and so on. So these drugs, even though they help, but they come with a plethora of side effects that have very adverse effects on the health. Some of the common supplements used include boswellia, green lip mussel, MSM or methylsulfonylmethane, and chondroitin sulfate.
Yeah, and I think, Prabhakar, 1 second. So I just want people to look at this slide. These are the current supplements that are out there. The question to all of you is, do they work? How do you quantitatively know they work? And you would have to go read thousands of papers here and there to piece it together as a consumer. Now, what we're going to share with you first is with CytoSolve, we can quantitatively and very scientifically at the molecular systems level figure out if they work or not. And we're going to walk you through that. And then we're going to walk you through MV25, how we created that. So go ahead, Prabhakar. Thanks.
So we have spent almost about 2 to 3 years developing or working on this area of joint health in collaboration with University Health Network in University of Toronto in Canada. And that resulted in a open for public a tool that allows you to take a glimpse at all the molecular mechanisms that are involved in the osteoarthritis, especially in the knee of the humans. And let me show you the final work that we have created with the University Health Network. So as I mentioned, this is a collaboration between us and UHN. So this is a visualization of
Prabhakar, one thing I just want to give a background. I don't know if you have that slide where we show the funnel, but what we've done here is we've looked at every paper written in the world on osteoarthritis, joint pain, every paper, about 100,000 of them. We whittled it down to 22,000 that were relevant. And then from those 22,000, we looked at the ones that are related to humans. And then from each one of those papers, we're extracting chemical reactions that are involved in different parts of joint pain, like A plus B goes to C, C plus D goes to E. So you're looking at a linked network of all these molecular reactions. And then we had to put it into a visual architecture that could be made accessible to the scientific community. So we gave this away to the world. This is what's called the systems architecture, and we can do this in any disease. And then we have done this as a part of our research and educational foundation at the International Center for Integrative Systems. So all of you, and we did this on our own. Imagine what we could do with all of you guys taking different diseases and wanting to fund it essentially for pennies or finding people. But you'll see the level of work that's been done here. Go ahead, Prabhakar.
So what we're showing here is the different tissues that are that comprise the joint of a knee. That include synovial tissue, cartilage, immune cells, subchondral bone, and so on. And each of these tissues has a set of cells in that tissue. So, for example, synovial tissue has 4 different types of cells. And then similarly, cartilage is another type of tissue which has only one type of cell called chondrocytes. So if we go into the chondrocytes, we have these ligands that actually land on the chondrocytes and initiate the molecular biological processes inside the chondrocyte. These ligands comprise of growth factors, cytokines, different molecules, so on. So if you look at these major cytokines, which are the ones that are responsible for progression of this disease, we have almost 15, 20 cytokines that affect the disease. IL-1 beta is one of the major cytokine that actually creates the worst symptoms of the disease. So IL-1 beta, if it lands on the chondrocytes, it upregulates or downregulates several molecules. So the ones that are shown in green are upregulated, and the ones that are shown in red are downregulated by IL-1 beta. So as an example, MMP13, it's a protein that actually actively degrades the cartilage, causing a catabolic effect, which worsens the disease. So when we click on that, you can actually go and see all the molecular pathways that IL-1 beta initiates, and that lead to the MMP13 and the catabolic effect. So each of these lines actually represents the information that we got from the existing research. So if I click this, it'll take you to the scientific paper from where we got that. So that's the architecture of joint health in the knee.
Yeah, one thing we want to show here is what Prabhakar just took you through. There's hundreds of thousands of these chemical reactions that are now integrated here. This systems architecture is now made accessible to the world. So any researcher throughout the world can say, hey, this is correct, or I don't believe in this, or they have a different experiments. So we've essentially become like the central repository for this. So this is what we call it open science. So if you do research, you can contribute, you can change it. But this framework can be used now. So we've discovered it, we've published it. And now the third step is to mathematically convert those small reactions into quantitative equations, which can be used for discovery. So Dr. Prabhakar will now share that part of it. Go ahead, Prabhakar.
Yeah, so from all those mechanisms that we shared, so these are some of the biological processes that actually have a significant effect on the joint health. I have shortlisted 6 of them here that include the arachidonic acid metabolism, PGE2 signaling, COX2, oxidative stress, cartilage degeneration, and regeneration. And each of these slide contains the explanation or the molecular pathway for that particular signaling. So we identified 6 of these these signaling pathways that directly impact the joint health. And then we looked at what are the biomarkers of each of these biological processes. What that means is that when you take any intervention, how do you measure whether that intervention is successful or not? So the way that we do it is by measuring these biomarkers. So for example, for this particular biological process, PGE2 is the biomarker. And similarly, there are 2 biomarkers for PGE2 signaling, and these are the pain-related biomarkers that we'll go into details in the later slides. For COX2 synthesis, COX2 is again an inflammatory enzyme that we saw earlier, which is being inhibited by turmeric and resveratrol. Oxidative stress is another biomarker that actively causes the discomfort. Cartilage degeneration, the MMP13 that we saw in our systems architecture, and then cartilage regeneration, which is collagen. So to improve joint health, we need to downregulate these biomarkers except collagen too. We need to improve collagen so that you have a better joint health outcome. So these are some of the commonly used supplements that Dr. Shiva just briefly mentioned earlier. But do we really know whether they work or not? That's a big question. So the way to understand it is that the way that we have understood is by using CytoSolve and developing the models for all these biological processes and looking at the effect of these individual molecules on these various biomarkers. So of these, as you can see, the level of green or the saturation of green represents the effectiveness of that particular ingredient. So of these almost 10 ingredients, there are only 4 or 5 ingredients that probably exert a pretty good effect on the biomarkers of joint health. So with the analysis that we are able to do, we can actually say which ingredient actually works better individually or as in a combination. So that leads us to how we actually came up with the MV25 formulation. So we saw that There
Prabhakar,
are
1 second,
several.
if you can
Yeah.
just go back to the previous slide on the, so I just want, again, people to recognize that this is the first scientific quantitative analysis of these other individual supplement ingredients people claim to have effects. So the key point here that you want to think about is when someone says, oh, there's a product for joint pain, joint health. I hope everyone understands that's a very, very broad statement. It can be misleading. The issue is joint health is not any one thing. Joint health, quote unquote, joint health is a function of multiple different molecular reactions that need to be dealt with for you to really have good joint health. Okay. You have to make sure inflammation isn't there. You have to reduce oxidative stress. You have to make sure your cartilage isn't degenerating, and you have to regenerate cartilage. As you can see here, these individual compounds only affect one or maybe two of those things on a good day, but typically they're affecting one, and even that, not that good. So this is a problem with the entire supplements world. Pharmaceutical industry has its problems because they are single compounds that create side effects. Here in the big vitamin world, people are just throwing crap out there, and it's all based on marketing. It's not based on any real molecular science. And so this is why a number of those big vitamin companies, when we call them and say, hey, we'd like to test your product, you want us to work with you, they get a little bit afraid because they know they're just riding their entire process on basically vaporware. So just look at this. We'll come back to this, but we are taking a whole systems approach to joint health. It's many, many different things you got to do. You got to make sure oxidative stress is low, inflammation is low, you don't destroy your cartilage. That sort of whole picture is joint health. That's the big elephant in the room, not just any one piece. Okay? So now Dr. Prabhakar will walk you through how we created MV25. So we have the architecture, we did the publication, we did the models. And how did we create MV25? Go ahead, Prabhakar.
So we follow a 6-step process for any of our solutions that come out of CytoSolve. The first step is creating a systems architecture, the one that I just briefly went over, of the joint, the knee. So that gives us the various molecular reactions that are involved in a particular disease. For example, in this case, joint health or joint pain. So once we have that, we also we actually publish our results, and then we at the same time we also identify ingredients that can have a positive effect on the indication of interest, in this case the joint health. So of these from these ingredients, we go into the scientific literature and we shortlist the ones that are most relevant, and then we create the molecular systems architecture. From that we create the mathematical model, and then we publish, and we create the product. So from this systems architecture and the ingredient identification, we identified 4 different molecular pathways. The arachidonic acid metabolism, which is basically the inflammation pathway, oxidative stress, COX-2 synthesis, part of the inflammation pathway, and the PGE2 signaling, which is the part of the pain pathways. And we identified 2 ingredients, 2 natural ingredients that come from citrus fruits, as well as leafy vegetables such as celery, these are parsley, celeriac, and spinach. And the hesperidin is from grapefruit, fruit, and bitter orange and lime. So using those 2 ingredients, we actually see the effect of these 2 ingredients on the biomarkers of 4 different pathways. So as one of the outcomes of this research was this paper that we published that analyzes how exactly apigenin and hesperidin individually as well as in combination have a positive effect on all the biomarkers of pain and inflammation. And I have some, this is where we do the mathematical modeling. So we have these 4 individual models that we created, and we incorporated the effect of apigenin and hesperidin in each of those models. We used the CytoSolve platform to combine the effect of these 2 compounds on the biomarkers of all these 4 biological systems, which govern the joint pain. These are some of the results. So the first few slides will have the individual effect of apigenin on each of the biomarkers. As you can see, for COX-2, at control, we had about 8 nanomolar of COX-2, and when we add the apigenin, it reduced to 2. So there's a significant reduction in COX-2 when we added apigenin. So similarly for PGE2, there is a significant reduction when we add apigenin for TRPV1, CGRP, as well as except for reactive oxygen species, because apigenin did not have any effect on the ROS. So that's why you see that the control value for ROS is very similar to that of apigenin. So the next, we looked at the individual effect of hesperidin. Hesperidin was also able to reduce COX, PGE2, TRPV1, CGRP, as well as ROS, significantly reduced the reactive oxygen species by hesperidin. So that's the individual effect. So when we combine, we find a much more pronounced effect in each of those biomarkers. For example, here, COX-2, if you remember, it went from 8 to 2 when we had apigenin, and 8 to almost a similar value. But when we add both, it actually reduces the value of COX-2 very significantly. Similarly, for PGE2, there's a synergistic effect there for both TRPV1 as well as CGRP, the pain biomarkers, and then also for reactive oxygen species. So while we were doing this, we also wanted to run another set of experiment to understand whether actually the apigenin and hesperidin are having any a synergistic effect or additive effect. So the difference between that is that if two ingredients are having an additive effect, you'll have kind of a linear decrease in those biomarkers. But if they are having a synergistic effect, that means 1 plus 1 is not 2, but actually it's 11. So there's a significant downregulation of that particular biomarker if there is a synergistic effect. So to understand that, in this particular experiment, we added apigenin first and then hesperidin second. Then we found what is the level of COX-2 after this experiment. After that, we reversed the order of addition. So we added hesperidin first and then apigenin second, and then we identified the COX-2 levels. So this gives us a range of additive effects. That means if you add the compounds simultaneously, and if your value lies between these two values, then you're going to have an additive effect, 1 plus 1 equals 2. But if it lies beyond or below that, then you're getting actually a synergistic effect. So when we added hesperidin and apigenin simultaneously, the value lies outside this range. So what these results show is that product that we have developed that contains apigenin and hesperidin actually has a synergistic effect in lowering the biomarker of pain, COX-2. So that's a pretty significant finding. So this is just one parameter. So the next parameter is PGE2. So here, our range for additive effect was 217 to 289. And when we added the compounds simultaneously, we found the value to be 186. That is between the additive range. So that means in this case, apigenin and hesperidin are actually having the 1 plus 1 equals 2 effect on reducing PGE2 levels. So after 5 parameters, we observed the synergistic effect of apigenin and hesperidin on all the biomarkers except PGE2. So for example, for TRPV1 also, there was a synergistic effect. This value lies way beyond or way below the additive effect. Similarly for CGRP and ROS. So for ROS, the additive range is 1.34 to 12.69, but our simultaneous addition reduced the levels of ROS to 1.11, which is lower than this range. So that means we were able to, or this combination is able to synergistically reduce the biomarker of reactive oxygen species. So comparing the heat maps, what we had just seen for the other compounds, apigenin and hesperidin between themselves are affecting all the biomarkers, and they are having a very significant effect on each of these biomarkers of pain. So it is that include PGE2, CGRP, TRP1 for pain.
One second,
ALK2,
Prabhakar.
and
I think,
yeah.
Prabhakar, 1 second. Important to let everyone know that this is all about the right combination at the right dosage. So when Prabhakar goes back to those slides where we're looking at the synergistic effect, you see the United States Patent Office, you can't simply read, the newspaper, go online and say, oh, I'm going to throw this and this together, give me a new innovation. You have to prove that something is synergistic. That means 1 + 1 is 10, not 1 + 1 is 2. When you show that, that is called a synergistic effect. So what we are showing here is that for these combinations across these different biomarkers, which are variables which measure particular dysfunction, that our combinations are out of the additive range. They're synergistic. Very, very important to remember, but it's at the right dosages. You can't just say, well, I'm going to put a little bit of this and a little bit of that. And this is what's going on in the big vitamin industry. If you go to people and say, why did you put that much and this much in? The reality is they're putting that much and this much in. Sometimes they think more is better, or that's what they could purchase at a lower cost. They're not really figuring out quantitatively what gives you the right combination for the biggest bang for the buck. So if we now go to that heat map, the important thing to note here is this is at the combination. Prabhakar, go to the heat map. So we're looking at specific combinations, and you can see that this combination is a very powerful effect. Prabhakar, if you go to now the other one, I think you have it right there. This is what those individuals are. So if you go to Whole Foods or Amazon, you're buying a lot of these supplements with chondroitin and vitamin C and MSM and yucca. And you notice these are nowhere near, previous slide, what with the combination MV25 does. This is the point that we wanted to share with you. We have quantitative data based on all those molecular reactions coming from all of the research integrated together. And this is not possible by any other platform. Thanks, Prabhakar. Next. So this is a key takeaway. This slide and this slide should make it very stark. And then furthermore, it took us 3 years to actually get a patent. What Amazon is doing right now, if you go to Amazon, you buy some product, they just watch what's selling and they just copy it, because all of these other products actually don't have any real innovation. The United States Patent Office gives stuff. So this is another proof point. Not only did we publish this, not only did we publish it twice, not only did we discover, but we also got a US patent, which protects us now for 20 years. So Amazon can't steal this. So a number of you have said, hey, I would like to sell MV25 in my health food store. You have a protection here because this is a product that was created in a decentralized way, bottoms up, by integrating all these reactions. So this is what we've done. And we didn't do this for hundreds of millions of dollars in research funding or 50 million, 10 million. This was done just through our sweat and hard work. Next, Prabhakar. So that's MV25. If you go to mv25.life, then we had to go create the website. Now we're learning how to sell it. We did it all on our own, right? So we've created this fully integrative platform, from research to modeling to combination screening. And this is not funded by VCs, private equity, big pharma, or big vitamin. This is really funded by, in many ways, this movement for truth, freedom, health. In many ways, this product is by the people, for the people, and will continue to. So one cool thing about this is, if let's say some new ingredient comes, we're going to see if that works. We can add it to this. If we find new research comes, so this is a work in progress, but we know quantitatively how it's doing. And we're fully transparent. Everything we've shared here, you won't find other companies even sharing this level of detail. We're transparent. It's bottoms up. It's integrating, hundreds of thousands of reactions we've published, et cetera. And you can't say this about 99.9% of the stuff out there. Next. So how you can help? First of all, go buy our product. This is first. We have another one we just developed for pets, and you can develop products through too. Fund our research. Go to CytoSolve and just fund us. I think go to International Center for Integrative Systems or go to Truth Freedom Health. It all supports this research. You can also become an affiliate. It's not a multi-level marketing scam, but you yourself can directly be an affiliate, and we support you if you want to do that. You can also produce your own products with this infrastructure. You can support indigenous medicines. If you are different parts of the world, there's some grandma's recipe, but you don't know why it works. We can help figure it out and help get it out to the world. And then the other thing is join the movement for Truth Freedom Health, because by joining that, we take a portion of that to support CytoSolve research. Next. So how you can help buy CytoSolve products? Well, Prabhakar, if you can go back to that. So, no, go back. Yeah. So think about all of these natural ingredients is where we discovered these compounds and then we did this analysis, and that's how we created MV25. So one is you can go buy this product. Next. We also have, you can go to mv25.life. We also just recently discovered a combinations of food ingredients, concentrated food ingredients. When you put them together, it's very valuable for your pet's overall health, and that's K9701. Next, and maybe we'll do a follow-up presentation next month on this. Next, you can help fund research. Next, you need to recognize that we want to support true indigenous medicines. We want to eliminate animal testing. We also want to fight the existing incestuous web of science. The same people who are funding the research are the same ones who sit on the pharma boards, etc. So you can go to vashiva.com and donate. Please do that, because we've shared a ton of information here. It's one way for you guys to support what we're doing. Next. The other piece is we have various research areas. Prabhakar, go back. And you can create your own research area. If you say, hey, one of my grandparents died of this disease, I want to go support that. You can go crowdsource, raise funding. You can become your own research institution with this capability. Next. And by the way, when you donate to us, we give you a lot of things back. We give you books, we give you tools. Next. And then finally, the systems architectures that you're seeing are hosted and supported by the International Center for Integrative Systems, which is a 501c3 nonprofit. Here you can donate and you get your tax write-off. Next. How you can help through affiliate programs. Next. You can become an affiliate to resell these products. You can refer products developed by CytoSolve, and you can refer companies. And if you want to become part of our affiliate program, next, you can literally go to vashiva.com in the customer service section, and there's an affiliate application. So fill that out. Next. And then you fill that out, and then you can help with product development. You can develop new products. As a part of doing that, from our last symposium, we had about 4 or 5 people who wanted it. And you can also use this to do research. Next. So you can simply, you can do your own product. And the way you can do this, you can schedule a meeting. Prabhakar, you may want to put that up. So you can schedule a 60-minute meeting with myself and Dr. Prabhakar and others, and we'll literally walk you through. So you should come with your idea of the intervention and then what area you want to go after. You can also do it for the other side, toxicity. Does these ingredients cause toxicity? So Dr. Prabhakar just put up mv25.life. That's a link. He's putting up the Calendly link. The Calendly link literally lets you go without having to go emails and literally schedule a meeting. So if you can put that up, Prabhakar.
I did, and I'm going to also put the customer service.
Yep. I think that's it, right, Prabhakar? So let's summarize. Everyone can see this, yeah. So there's the customer service form. Thanks. And you choose the affiliate application. All right. I think in summary, do we have a summary slide?
Yeah, we still have to go over the.
So in closing, everyone here, if you made it this far, become a Truth Freedom Health Warrior Scholar. It's a way you can support what you're doing and you support yourself. Go to truthfreedomhealth.com. Prabhakar will put that up too next. Towards the end, we can put up all the links. You can come to our next tutorial. I think if you make Emily Prabhakar co-host, Emily will put up all these links in one shot. Actually, I'll make Emily a co-host. Emily's got a nice email. I'm going to make Emily a co-host. Yeah. Emily, if you can just put that up to everyone. So Emily's going to bring in a bunch of links, but we have our systems health tutorial coming up. This is for those of you who want to basically become a systems health educator. There's a business model here. You can get out of maybe some unsatisfying job, or you can just help people. Next. Again, in closing, we're about thinking, fighting, and healing. We have a very powerful set of platforms. So you basically have no excuse to say you can't do something. You yourself can do something through these very powerful platforms. Next. That's it. Let's take Q&A. It's 12:28. Let's take Q&A for 15 minutes. And then we'll wrap up. So we've had a good number of people. We have 93 people. Double what we had last time. It's good. So in order to ask a question, there's a reactions button on the bottom, and you can click that. And then the thing says raise your hand. And then Dr. Prabhakar will unmute you. In the interest of time, if you can, keep your questions tight, that would really be valuable for all of us. And then we'll attempt to answer it. But again, if you want to have a follow-up meeting, use the Calendly link. If you have an idea that you want to build, develop, research, become a Truth Freedom Health Warrior, that should be sort of a no-brainer for everyone. You have to become part of that, because without understanding politics and the intersection of truth, freedom, and health, you're going to be seriously handicapped. Any questions? Okay, so let's start with Sonia Frederick. Go ahead, Sonia.
Hi, Dr. Shiva.
Hi, Sonia.
A gentleman online was basically accusing CytoSolve of being part of the transhuman agenda. And he, I wasn't able to really answer him because I didn't understand completely how it all worked.
I saw that guy. He sounded like a fucking idiot. I didn't even know what he was saying. He doesn't understand
he's saying that the information that you're gathering is coming from biomarkers that are inside of the human body.
No, he's an idiot. He's a bloody idiot. You see, the problem is you have these fools. It's a complete idiot. I don't even want to. This guy's an idiot. He doesn't even know what a biomarker is.
Yeah, I wanted to be able to answer him and just,
You can just say you're an idiot. Look, certain people are just so outlandish. It's just a fucking idiot. Excuse my language. I saw that and it sounded like a blithering idiot.
Yeah. Well, I don't know a whole lot about all of these symbols, and I get confused, so I didn't want to answer him and make the situation worse.
No, he's just making up nonsense.
Okay.
The problem is we don't have people at the grade school level who are learning chemistry anymore. We don't have people understanding what a chemical equation is. So the internet is polluted with idiots. We're just making up shit because they want views and clicks. Ask him, does he even know how to add a number together? Ask him if he knows what a mole is. And it's not a thing that's running around in your house. It's molarity. So you have a serious problem I think it starts around 5th grade now. 5th, 6th, people aren't learning basic chemistry.
Right.
So biomarker is, sodium plus chloride gives sodium chloride. That's a chemical equation. The level of salinity in that case is a biomarker, you say? So these guys are, I saw it, that this guy's an idiot.
I mean, the simplest way is when he goes to get his blood done, when the doctor tells him that, oh, your sugar levels are high or your cholesterol is high, that's exactly what the biomarker is. Your sugar level, your cholesterol.
H1AC levels, yeah. So if I take blood out of you and you're looking at different chemical compounds, this guy's a complete fool. I mean, you guys need to recognize that what's happening right now is there's the elites know that when guys like me expose the real stuff, they're creating disinformation the other way, Sonia. You say? Just throwing out whatever will stick. And none of these people they haven't put in the work. They don't do the work. They just talk nonsense. But anyway, as Dr. Prabhakar said, a biomarker is you draw your blood, you give it to a lab, and they measure the levels of chemicals in your body. So if you have a high level of XYZ, that's indicative of XYZ condition. You follow? That's what a biomarker, it's just a chemical. All right, next. Let's go to Jasun. Jasun, go ahead. You have to unmute. Jasun, could you unmute, please? Okay, no. Let's go to Shahnaz Khan. Unmute. Do people know how to unmute? Hello? Prabhakar, are you there?
Hello, I've unmuted. Can you hear me, Dr. Shiva?
Yes, go ahead, Shahnaz.
Oh, hi, Dr. Shiva. I'm sorry, I haven't been in touch for a while. Excuse me, shush. I haven't been in touch for a while. I've just rejoined your course, and I think the stuff you're doing is phenomenal, and it's amazing. And I work as an alternative health practitioner, and I'm based in London. And what I'd like to know, the MV25. How can
Yes.
I get hold of it in the UK? And I also want to, test it out. I want to test. I don't know if I can test it out amongst my clients, but I also want people who are ready to start taking it. So what's the process I need to follow in order to get
right
hold
now,
of it?
Prabhakar, so we'll connect you up with Ken Fielding. So Prabhakar, if you can connect Shahnaz, Prabhakar is going to text you right now. If you can give your contact information, if you're on WhatsApp, your phone, and
Yes, I
your
can.
email, Ken will get back in touch with you literally within the next few hours and help you out with that.
Fantastic. Okay,
Yeah.
Brilliant. And thank you so much.
Yeah,
we
and I
love
think
you
those
from
of you who are in
London.
different parts,
Yeah.
those of you in different parts of the world, we spend a lot of time on development, not on the business side of the selling. It's only recently we've done this. So we realize the solutions we create are so innovative and revolutionary that because they go against the establishment norms, it would take us decades to do the typical model where you raise money from the establishment, you have to sell out. So our future is only going to come from you guys, like Shahnaz, what you said. So if you're interested, we want our network of all of you to become our extranet of people who we want to support. So Shahnaz, it's so there are chiropractors, there are alternative medical people, there are some of the more thinking doctors. MV25 is perfect for them because you already have a trusted relationship, and this product is magnificent. It works, and people value you for it, and you can explain it. You can become an educator through this product on many things, like we just did.
Well,
So
my teacher's based in Malaysia, and he's the one, and it's the Malaysian royal family who actually are the ones who told us about you, Dr. Shiva. And I've been, since lockdown, I've been on and off following you. But even without even testing your stuff, my teacher, he's like a Sufi kind of lecturer, and he has students globally, and he really endorses you, and he talks very highly of you, and he really trusts you. So even without trying the product, I'm so desperate to get hold of it so I can try it in London, because I believe that what you do, there's sincerity in what you do, and there's genuine care and truth. And my teacher was saying, Shiva really is out to help people. He's on a mission to help people. And I think that's key for people to understand, that this is not a money-making drive. And if you do make some money, why not? You're doing something for the good of the world.
Well, Shahnaz, you're bringing up. So anyway, give me the name of that person, because I was in Malaysia because one of my close friends is a neurosurgeon there, and he had mentioned something like this, and I didn't know what he was saying.
Oh, you've got to get in
I,
touch. He's absolutely phenomenal, honestly.
yeah,
I mean, he's the one who's getting us all. He's talking about you to all of us. He's the one who's encouraged us to go on your course. I mean, I've been rubbishing being in touch
wow,
because of
okay,
personal reasons. But I signed up to your course blindly in 2000 when it was lockdown. I didn't even know anything about you. But
right,
you are very, very popular amongst our group of people, and we've been trained in alternative medicine. There's doctors in UK, trainee surgeons who are wanting to give up their work because they know that what the pharmaceutical industry is doing. So once I get your product, I'm going to market it here, and I want to encourage people in my field. And there's loads of doctors
okay.
globally that we
Well,
want to, yeah.
well, Prabhakar will help you. So two things, Prabhakar, if you can chat right now to Shanaz directly, Prabhakar, if you've done that, and Prabhakar, can you
Great.
get the information from Shanaz?
Yeah.
Prabhakar, are you
I'm
doing
very happy to share. Yeah,
Yeah.
okay? The
that's
other
great.
thing,
Thank
Shanaz,
you.
to broadly to everyone listening, Prabhakar, if you so CytoSolve is one of the platform solutions. The other platform is Systems Health,
That's
which
right.
it's a platform to it's a home for those MDs, nurses, not only for them who want to really recalibrate themselves. It's also a home for alternative health people, and it's a home for anyone. Prabhakar, if you can bring up that site. So if you go to Prabhakar, will put up that link again. But
Which one?
the Systems Health, Prabhakar.
Yeah.
So Prabhakar will bring it up, but this May, end of this May, we're having a Systems Health workshop. Now, what we've done is we've created a three. So we have essentially, I used to teach this all over the world at MIT, etc. But we've created a certification program. It's called the Systems Health Educator Program. Prabhakar, can you bring that site up? And what you notice is that the Systems Health Educator Program involves an entire like training program. So you go through, there's hundreds of hours of online courses. You learn the integration of Eastern and Western medicine at a very deep level that integrates engineering science. So as the cover says, Systems Health Institute is beyond health coaching. You can get it online or in person. I love doing it in person, but some people can't travel. And it says, what is this? It's an educational system and a technology platform enabling you and your clients to navigate contradictory and confusing recommendation prevalent in the health and wellness industry to uncover the right solution for the right person at the right time. Scroll down. And the core of this, Shanaz and other people may know, is this. I uncovered this knowledge when I discovered the connection between engineering systems and ancient systems of yoga and medicine. So, Prabhakar, if you scroll down a little bit, and I encourage you to go through the site, but we have done, I used to do this in 2012, 2013, 2014, 2017, and then we put it on stasis because as I was getting involved in politics, electoral politics. But we relaunched, we've done 2 workshops, and a lot of people gave us feedback. Some people said, you're not charging enough, believe it or not, Shanaz. So, but we want to support all of our Truth for Health people. Some people needed ways to make it financially accessible. So we've done all that. We really listen to people. But if you go down, this really explains. So just like CytoSolve is a technology, Systems Health. So this is so the 3-day summit right there, Prabhakar, stop, is part of the courses and the tools. But we actually give you a technology that you can use to help your clients out or help yourselves and your family figure out what's the right solution for the right person. But in the 3-day summit, which is an additional stuff, we go through the systems healthcare. We go through what is the source of Western medicine, what is systems biology, what is engineering system science, what is Ayurveda. And as I mentioned, those 4 are completely different disciplines: engineering, biology, ancient systems of medicine, system science. And then we bring it all back to a systems understanding of what is real health, the concept of resilience. That's day 1. Day 2, we do an entire laboratory where we introduce you to this very powerful tool, the concept of circadian rhythms. We teach you a very powerful, potent way to meditate and do yoga and what is yoga. And then we provide you a capability to create personalized products. And we have some of those coming out. And then the last day is really to help those people who want to create a quote unquote educational platform or educational business doing this. And then you can go through the whole thing. We've made it accessible to everyone. But that's the Systems Health program. Thanks, Prabhakar. But anyway, that's another solution. So, you know, to the extent that I do a lot of, I unfortunately have to use the most powerful vitriolic words to expose the snakes out there. But in 2024, we're deploying all these amazing solutions. But thanks, Shanaz. Thanks for your kind comments. But please let Prabhakar know so we can connect with those people. Let's go over to
Dr. Shiva, just I'm back on audio.
Yes, Justin, go ahead. What's your question?
Oh yeah. Hi, doctor. I have a severe osteoporosis. To make something for it, I'm taking MB25. I'm dosing
Well,
up
yeah,
30.
so I know you sent me an email on this. So this is so osteoporosis. So think about what Jasun is just asking.
Yeah.
In the interest of moving people, I'm going to mute you and I just want to go through that question. But Jasun sent me an email. She has osteoporosis. So osteoporosis is a particular condition where you have deterioration of the bone. Now, what we have, so if you just look at what we have the capability to do, Jasun and everyone, we have not created the architecture of osteoporosis, but we can. So we can literally create a model of osteoporosis. Then, just like MV25, we can go find those combinations of ingredients which can support it. So what we just shared with you is one example. Prabhakar, you may want to bring up the NutriSolve slide of all the different things we're doing, but Jasun, if you're interested, and others on this call, if you're interested, imagine you, all of you as citizens, how would you get research done on osteoporosis? So just think about how would you do it? You can't. That's the answer. Or if you did, you'd have to go on PubMed and read articles all day long, and you're never going to get anywhere. So we as citizens with this enabling technology, we will help you. We can do a meeting if someone wants. That's why I put up that calendar schedule. We can show you the actual step one, step two, what we would do. We can, if you try to have a university do this, they've spent hundreds of millions of dollars. CytoSolve can do this at 1/1000th of the cost and actually make headway in anywhere between 6 to 18 months. And all of you can fund that research, or you can create a consortium, crowdsource it, and we can together discover a solution. Everything we do, it's transparent. As we're doing it, we do meetings. We let you know how we're doing it. You don't get that from when government funds research. So Jasun, that's the answer to it. But osteoporosis affects a lot of women as they age, right? So I recommend if this is an area you guys could do it, we're here. We have a way to do that.
Yeah, I'm taking MB.
Let's go to, so anyway, the action step, Jasun, is click on that Calendly link and you can set up time if you want to gather other people or other people want to do it. Let's do that.
Okay,
Ellie Kelly, go ahead, Ellie. Ellie Kelly.
Okay.
Unmute, please, Ellie.
Okay.
We'll go to Ellie, then we'll go to.
Am I unmuted? Can you hear me?
And Betty Plummer, then Griffiths Hamilton, James Payette, Eric, Bernardo.
Can you
and
hear
Bonnie.
me?
Let's go, Ellie.
Okay, can you hear me? Can you hear
Go ahead.
me?
Yes,
Yes,
go ahead.
we
What's your
can.
question?
Okay, I'm a veterinarian. I have two questions. Have there been any studies done on interactions between NV25 with different pharmaceuticals like NSAIDs? Because a lot of my patients are on NSAIDs. And if I switch them over, do I have to cold turkey them off of NSAIDs to get them on NV25?
Yeah, so let me just, so when you say NSAIDs, you mean NSAIDs, non-steroidal, yeah. So like ibuprofen, etc.
Right.
No, we have not, but I can tell you from a veterinarian standpoint, this product, after we first, came across it and put together all the work, it has been tested with horses, equine, okay, with profound effects. We have not done the interactions with it, but I would say one of the things we did many years ago was looking at other natural anti-inflammatories with ibuprofen, for example. what we found was you could reduce the dosage of ibuprofen significantly.
Okay,
So,
okay,
so it doesn't, because remember, ibuprofen is a COX-2 inhibitor.
right,
So this actually hits COX-2 pathways. Okay. So it's not going to be antagonistic to it. If anything, it'll be additive to it. So you can do less of. So we've had a
Okay.
number of people write to us, and unfortunately, you know, you need to do clinical research, which takes money, right?
right,
Which we can do. But you know, if you're a vet, we
right,
could do an IRB with you, and you could do this in your um in your practice, right? You could have people or you know, with animals come in, you can give one of them NSAIDs and you can give them, we could do an IRB test if you wanted and publish it. But it is not going to have a negative effect. It'll, if
Okay.
anything, reduce the amount that you need to take of the existing. So we've had a number of emails. Oh, you know, I was taking 2 to 3 ibuprofens every day. I don't have to take any of it. I was on. Now, I don't want anyone to take that as medical advice because that's where the FDA will get us in trouble. Okay. I just want to let you know
Okay.
this is what we've heard anecdotally. But we could
Okay.
do. But the
Okay.
bottom line is MV25 hits 5 major pathways, one of them being the COX-2 pathway. But
Okay.
ibuprofen, that's all it hits. Okay.
Okay, but I was, there's nothing about potentiating liver toxicity from ibuprofen or something like that. It's not going to do anything like.
I look, what I, you know, I don't want to, we haven't done that work,
Okay,
but
okay,
what I want to be careful to say from the efficacy standpoint,
yeah,
remember, these are 2 natural compounds.
okay,
One comes from chamomile and the other comes from
okay,
bitter orange. Okay. They're
Okay.
2 bioflavonoids.
okay,
They are generally, you know, referred as safe, you know, grass.
yeah, right,
So,
okay,
and the dosages we've done are far below, you know, I mean, they're like so relatively low. It's a synergy of them.
okay,
But it would be, I mean, what we could do, Prabhakar, I mean, we could do this if someone wanted to, we could take ibuprofen and throw it into this and see how much little less of ibuprofen you would need. You know what I mean? Like we
yeah,
could see if you're taking 200 milligrams of ibuprofen, maybe you only need 1 capsule of this versus a 2. You see what I'm saying?
right, right.
That's what I'm saying.
All right, and my
Or-
other question would
Yeah.
be, what, from the time you get on this, when do you see any kind of effect? I mean,
Yeah,
like,
obviously. Yeah. So we have Prabhakar, we'll play a quick video so that'll give you different people's experiences. So obviously people have chronic. So one of the interesting things is the apigenin also crosses a blood-brain barrier. It's one of the things that out of all the Alzheimer's stuff, it's been one of the 3 compounds that they really believe is promising. The reason I shared that it supports in the vasculature, you say?
yeah,
So when you take, so typically, there are people, there's a construction worker, we'll play his testimony. He was taking it. He said it didn't help me right away, but eventually it helped him and all of his back pain went away. Okay.
okay,
Like it's not like ibuprofen, but meaning but if you take the dosage at like 3x what's there and it doesn't harm you, it has much more immediate effect. Okay. So
Okay.
2 is like for chronic pain, right?
okay,
But if you take 6, it has much more immediate effect.
okay.
Yeah. So Prabhakar, maybe it's a good time just to play that video very quickly. But anyway,
People
yeah, go ahead, Prabhakar. I'm sorry. Go ahead.
suffer every day from painful discomfort and swelling, but most pain medications come with harsh side effects, and many alternative supplements have little scientific backing. That's why we at CytoSolve created MV25. MV25 was formulated using the CytoSolve computational systems biology platform, a technology for precision and personalized health invented by Dr.SHIVA during his doctoral research at MIT. This formulation is the result of computing trillions of potential combinations of biomolecular interactions derived from thousands of peer-reviewed scientific papers published across 4 decades by 68 research institutions to discover an optimal synergy of compounds that downregulate biomarkers of discomfort and normal swelling.
Hi, I'm Barbara Ann. My hands would cramp up so that I couldn't hold cards or knit or crochet. and they would go like that. And I'd have to use this when I played cards with my grandkids. And I started taking that MV25. After a bit, I was able to hold cards in my hand. Very, very little cramping, hardly at all anymore. MV25.
Hi, my name is Sandy. I'm a Taekwondo instructor. I tore my ACL during Taekwondo. I had a lot of pain and limited mobility. I've been taking the MV25 for about 6 months now. After the first week, I noticed a big difference. After the second week, almost literally no pain.
My name is Jeremy, and I suffer from a lower back problem. Hurt my back at work years ago, and I can go to the chiropractor, do all kinds of different things, and nothing seems to help. And I decided to try MV25. I didn't notice a difference immediately, but within a few days, the pain went away, and it stayed away. I've continued to take it, and even when I do things that I shouldn't do, it seems to go away a lot quicker than it ever did before.
MV25 is certified clean, 100% non-GMO, made in America, and GMP certified for good manufacturing practices. MV25 is CytoSolve optimized, which means that this formula has been engineered to maximize benefits while minimizing toxicity based on current research curated by CytoSolve. As the science advances, so will this formulation. This is our promise. Order online at mv25.life. Consult your doctor before taking any supplement or medication and use as directed. MV25.
All right, let's go to Mukibi Robert. Mukibi, how are you?
I'm okay. How are you doing? This is I am from Uganda.
Oh, very nice to have you.
The pleasure is mine. I was introduced to you by my friend Segawa Arthur. I have 3 grandmothers' recipes, and they have worked very well on people with wounds that come from diabetes. And I also have another one that has helped people overcome pain. Their blood pressure goes to normal in the next probably 2 weeks. People with a bit of heart diseases, they get a bit of relief within a short while. I am now very concerned. How do I submit samples for you to help get the content of those grandmothers' recipes? Can I submit in a link? Must I export them to the US for analysis? Please advise.
Yes, so Mukibi, the first step that should happen is Prabhakar will send you a link. Let's schedule a meeting online, okay, where we can understand these 3 things. And then we'll describe the process we go through, okay? But what Mukibi you're asking is one of the missions of the Open Science Institute, because there are formulations in all different cultures where people have these grandmother recipes. And they've people have come to these from thousands of years of knowledge passing from one person to another. These things run through CytoSolve, and like we say, we can do the publication, patenting, and all of that. We'll be able to bring some amazing medicines that work to the public. And what we've done is we've eliminated academia, we've eliminated big pharma, we've eliminated all these institutions. It's from the people for the people. So in order to do that, Prabhakar's putting up the Calendly. Go to there and schedule time and put your Prabhakar, get Mukibi's email and phone right now. So then we will also, if you don't do that, we'll follow up and set up a meeting. So that's the first thing that needs to happen, Mukibi. Okay?
I'm very humbled.
Well, nice to have you. But that's part of why, I created CytoSolve, because I came from that background in traditional Indian villages where there were these formulations, and many of these ancient formulations are just being lost. So if we can figure out why they work, how they work, and preserve them, it truly is medicine by the people for the people. So it's pretty exciting. Yeah. All right, good to have you, Mukibi. But Prabhakar will get your information right now.
Okay, thank you very much.
All right, thank you, Mukibi. Let's go over to Betty Plummer. Go ahead, Betty. Betty, go ahead.
Unmute. Taking my
Hi, Betty.
call.
Yes,
Hi.
Betty, we can hear you.
Last year, my bone and joint doctor told me, well, your collagen's nearly gone in your knees. Once it's gone, it's gone. Can MV25 help me?
So MV25 can, so if you looked at those pathways, Prabhakar will bring that up, okay? Prabhakar, if you bring up those pathways, remember there are four things at minimum involved in joint health: inflammation, oxidative stress, right? Cartilage regeneration. Oops, someone's, I got it. Hold on. Someone is.
Thank you very much, doctor.
Someone's okay. So to answer Betty's question, there are. Wait a minute, who's talking here? Hold on. Okay, there. So Betty, if you, and to others looking, Prabhakar, go to the previous diagram with the heat map, that there are multiple pathways involved, not this one, Prabhakar, the one above that. Yeah, right there. Yeah, right there. So Betty, if you see there are these different pathways involved in joint health: arachidonic metabolism, PG2, COX2, cartilage degeneration, and cartilage regeneration. But in your case, if you want to arrest cartilage degeneration, MV25, because all of these things are integrated, can obviously, in our view, be beneficial. To regenerate cartilage, you need something else, okay? Collagen itself, collagen 2, like hyaluronic acid. Prabhakar, if you go to the previous one, remember our goal here was pain and discomfort. Out of all the things that are out there, the hyaluronic acid, and we're going to be offering that soon, has effects. But regeneration is a little bit difficult to do because you have to, and that's why people are looking at things that support your stem cells to regenerate. Prabhakar, do you want to add anything to that? Prabhakar? Hello? Prabhakar, we can't hear you. Maybe you're muted.
Sorry about that. So most of the things that, including medicines that you take and supplements you take, are to stop things from going bad. And there are very few interventions that actually help you grow that is gone bad. So I think, as Dr. Shiva mentioned, the MV25 is going to help you prevent the further degeneration rather than improve the cartilage. But there are some molecules like hyaluronic acid, which is basically, it's a part of the cartilage. If you take some, and we'll be coming up with a product that contains hyaluronic acid shortly, that can help with the regeneration of your cartilage.
Now, there are certain things that we'll talk about more from a system standpoint overall that can potentially be very, very rejuvenative, that ancient traditional medicines have talked about. Because these cells, remember, it's the chondrocytes. Your cartilage is made up of chondrocytes that basically don't produce chondrocytes. The degeneration is occurring from the existing things breaking down and new stuff not being born. So on the one hand, you can, with the right environment, you know, food, lifestyle, all these things, you can start promoting growth also. But the goal of this is to support your normal, inflammatory processes. But we are looking at hyaluronic acid. That's something that we're literally working on, Betty. But MV25 will help arrest what's going on. Okay, let's go to Griffiths Hamilton. Go ahead, Griffiths. Griffiths Hamilton. If you could unmute kindly. Hi, Griffiths, go ahead.
Right. So, Dr. Shiva, thank you for that. I actually spoke to you, I think, on a previous systems health, one of the other ones anyway. But I've been researching toxoplasmosis for quite a while. Just wanting to know what sort of, it's because there's so many things involved with congenital, acute, and chronic toxoplasmosis. Is it something that you would, sort of think would be of value, or something that you could work on? It seems to have.
Yeah, I mean, that's the thing, Griffiths. We could work on anything. Where we are at right now is, Prabhakar, you can bring that one slide up on the NutriSolve. We're going after a whole bunch of areas that are in different stages of development. The reason we wanted to do this conversation is there are many other areas that we could go after. It needs public support. What do I mean by that? So if you wanted to go do toxoplasmosis research wherever you are, you'd have to, how would you do it? Right? You would have to give it, right now people give their tax dollars to the government, then the government decides if it's going to fund it. And typically the money is going to some institution that you don't even know. Next slide, Prabhakar, the one that we have all the different areas that we're doing. Yeah. So these are, if you can see, Griffith, these are the different areas that we're going after right now at varying stages. And we're doing this on our own. So pulmonary, rheumatoid arthritis, lung, stem cells, ALS, asthma, lupus, leukemia, right? Diabetes, dengue. There are people in Southeast Asia who wanted us to look at dengue, bowel, muscle health, gut, brain, pancreatic cancer, osteoarthritis, but we could easily add toxoplasmosis to this. Right. So what that would mean is the first step we would do is we would do the architecture. As many people know, when women are pregnant, one of the areas is they have to be concerned if they're around cats, which have feces, right? Or undercooked meat, because that causes toxoplasmosis. So what we would do is we have, interesting enough, we have done a number of pathways in this area having to do with infection, you see? So we could start there and go with it, but to let everyone know, the way the Open Science Institute is planned to run is that it has to have a champion, a citizen. So let's say, Griffiths, you're the champion in toxoplasmosis, right? We would then meet with you, and then we would do so you can link on the Calendly link Prabhakar put out, and then we would say, okay, this is what toxoplasma, this is the path. And then we would figure out what is the budget we need to do that. But our budget is going to be like literally 1/1000th. And then let's say so we would literally put toxoplasmosis as an area. And there are probably people out there in the world community who'd be interested in this. Sure. And we could figure out how so basically you become like an Oxford. You become like an MIT. You yourself, Griffiths, we would show you how you can go raise, maybe pennies, dollars from a whole bunch of people. And then we would set up the research plan to do that. So you become the resource development and then, CytoSolve has a platform to do all the work. So that's what I would consider a next thing to do. Right. But yeah, we could take on pretty much anything, right? So this is caused by a parasite, right?
For sure,
Dengue, malaria, right? It's a little bit different. Yeah. So we have had a lot of requests when I was out in Southeast Asia, in Malaysia, Thailand, people wanting us to take on dengue, which we started and you can see we're at stage 4 of this, so we can use some of those molecular mechanisms for this.
That sounds great. Yeah, no, certainly.
Yeah, but basically, Griffiths, if you want to do it, we're here. You can take the bull by the horns and drive it. So I would say the next step is set up time, like we just talked to Mukibi about, or we talked to, osteoporosis, Jen Soon about. So we have 3 projects right here. You can see osteoporosis, right? I think we talked about blood pressure and now toxoplasmosis. but literally you guys can be a part of enabling this to happen. Okay.
Yeah,
great.
that's good. I've also done some marketing and sales, so certainly interested in the in becoming an affiliate and that sort of thing as well.
Oh,
So
okay.
yes,
So
have a
what
good
you
chat.
do is, again, Prabhakar, get Griffith's name and phone number. Prabhakar will reach out, and then we'll connect you up with Ken Fielding. But it'd be great. Look, we really want to. We are not experts at. It's a whole nother effort in developing something and then getting it out there. And this is why we'd rather support you guys to help us get it out there, and it becomes very symbiotic relationship. So, Prabhakar, if you can get Griffith's name. Let's go to Eric Goodhart. Eric, how are you? Then we'll go to Bernardo, and then Bonnie, and then Garrett. Eric, how are you? Eric Goodhart.
I'm well.
Good, Eric. Where are you out of?
I'm in near Amherst, but I want to enroll. I want to get down to Cambridge on the 2 out of 3 days. I think I can do it. Is it necessary to have a laptop?
Well, if you want to physically come, you can come down. You don't need a laptop. You can, but come on down, because so by the way, the Systems Health program, there's a full-blown program where you can become a complete educator, but you can, I totally forgot to mention, you can just come for day 1 or day 2, okay? Or both. Let me just bring it up since you're bringing it up. If you're on the website, I think you've probably gone to the website, right?
Right. I was looking at that while you're answering questions.
Yeah. So let me just, since I know there's a lot of interest here. I'm getting individual text messages. So let me bring this up. So everyone understands pricing, costs. We spent a lot of time in making this very, very accessible to everyone. So let me share my screen here. Prabhakar, I'm going to start sharing my screen if that's okay.
Mm-hmm.
So if you go to the Systems Health site, Prabhakar will send you the link again. So the entire program is the Systems Health Educator Program. And what that includes is it involves 2 pieces, a system of education. So you get a 3-day pass to come to the summit. You can come as many times as you want, 4 textbooks, the paper, the lectures, 100 hours of courses, and then there's 5 certifications. Then we do monthly business mentoring for people who want to start their own educational business. Unlimited times you can come, and then a 12-month license to the educational materials. And then there's the technology. And then the way we priced this was, people said, frankly, you're charging too little. So we priced it at $8,000. But then there are people, if they're Truth, Freedom, Health warriors, because we want to incentivize people to become whole citizens, they get almost a $3,200 scholarship. And then other people said, wow, that's money that I want to do, but I don't have money to spend like that up front. So then we said, okay, you can pay it over time. And everyone's loved this. And we don't charge any interest. We don't want to be a bank. We don't want to practice usury. So some people can pay it up front. Other people want to do it without interest. And then we said, if people sign up today, and if there are people who just need a few more days, we may extend it until the 15th, because we've been getting a ton of requests because the email went out a little bit late. Yeah. But we can extend that. We may just change that. But if you do, you get another, because some people physically want to come into Cambridge for the 3 days. So we also said, okay, we'll include another $1,000 scholarship. So if people do want to do that, then it's $3,840, and it's a little bit less. Now, if people aren't ready to do that, they can just come day 1. Again, if you're a normal person, $950. If you're a Truth from Health member, it's $475. An early bird is $380, or 2-day. So we made it. Again, this came from hundreds of people's feedback. So we made it sort of mix and match. Some people can afford money. Some people don't want to be a warrior scholar. Some people have a lot of money in their lives. Money's not an issue. Other people don't. So we included this payment plan, and it's really on the honor system, but right when you sign up, you get access to everything. So that's what I would do. It'd be great to see you here in Cambridge.
I'm looking at Saturday and Sunday. I can't, prior commitment on Friday teaching. So
the way it works is, yes, if you come, You can, so if you sign up for it within a year, you can again, so if you come, so day 1 is Friday, day 2 is Saturday, and day 3 is Sunday. So if you sign up for the whole thing, you can just come for day 2 and day 3, and then you can come catch up day 1. Everything we've made also accessible online now.
Right. I can do Friday online, but
exactly. And then they can come into Cambridge for day 2 and day 3.
Yeah, that's the plan. Yeah.
Yep. Yeah, it'd be great to have you. Yeah,
that's good.
Yeah. But the Systems Health Educator program is to literally create global systems health educators. And then we teach you how to do a systems reset using the technology where you can meet with someone, figure out where their body should be, where it's off course, and help them figure out what's right for them. The health coaching industry, by the way, is going to become a $30 billion industry, but it's being inundated by the not so obvious establishment.
Yeah.
What we're doing here is giving you the vessel so you can really guide people with a framework, because all they're doing is just vomiting all these different ideas, Reiki, energy medicine, Ayurveda. No one is teaching people what's the right thing for them. And that's what we're teaching people, that infrastructure. Anyway, look forward to seeing you.
All right. Thank you.
Let's go over to Garrett Burns. Go ahead, Garrett. Garrett Burns. Garrett, can you unmute yourself, please?
Yeah. Can you hear me?
Yeah. Hi, Garrett. How are you?
Good. How you doing?
Good.
So I was actually, like, my family has a lot of issues with, like, I have all sorts of food allergies, like corn and gluten, celiac, and my mom's got arthritis, like rheumatoid and lupus and stuff. So I was wondering, it sounds like that's kind of what you guys are working on to try to figure out what the root cause of all that is.
Yeah. So on lupus, we spent about 3 years. We mapped out every pathway in lupus. We did it with, and obviously we did it with one of the researchers over at Cornell. So that's about to be coming out. But fundamentally, Garrett, we're going at the root cause of the many multiplicity of things that are involved. Now, ultimately, Garrett, it's food and lifestyle that's really going to do it, you see? But as people are getting their act together, these are very powerful vehicles, right, to support that, so people aren't suffering.
Yeah, because I mean, like you said, you're kind of just throwing darts in the dark or shooting a gun in the dark, trying to figure out what the exact issue is, and most of the supplements are crap. So, yes, it's nice to have kind of like even blood tests are garbage off the time. So it's like trying to figure out exactly what the issue is, what you should and should not be eating. Apart from doing like a water fast and for weeks and then introducing other foods periodically, then it's kind of a
if you go to, so one of the capabilities we have is in the ancient systems of Indian medicine, they always tweak the right medicine for the right person at the right time. So, Prabhakar, if you go over to, Prabhakar can just share the Your Body, Your System capability. So this is on the systems health side, where you can literally do a systems reset. So Prabhakar, if you go to Your Body, Your System, there's a couple of nice 3 diagrams there. The first diagram on top, Garrett, helps people understand through a series of, it's really an engineering systems approach embedded with ancient systems of Siddha, where we have honed in the questions that helps you figure out, go down, Prabhakar, what is your system type? Most people don't even know where their body's at because they're so out of touch with their bodies. The next thing lets you figure out what's going on in your body today. So that's the red dot. Now, the theory, underlying theory is your body is self-healing. So the black dot is where you're at. And the black dot may move around based on, how you've taken care of your health, what you didn't do. The goal is to, if you can get back to homeostasis, your body gets to be a resilient system. And this tool helps you calculate that. Now, we gave away this tool. We're always overly generous. I give away a lot of stuff. The problem is when you give away stuff, the human psyche, unfortunately, it's polluted with
the modern capitalist model, everything you charge for, so people don't value it. So we've had to figure out a way to charge for things that are still affordable. So your body, your system tool now, this is called a systems reset, is going to be offered through our systems health educators, because we've learned it's good to have a personal touch. Even though we have the self-service tool, we don't want to make this like, you know, just computers. So the systems health portal allows the, and by the way, you guys come Friday, we'll talk more about this. I don't want to take away from CytoSolve today. Allows people to figure out where they're at, where they should be, and then really focus on the lifestyle. And then we are training our educators in the overall educational model, plus we give them this tool so they can help as many people as they want. Let me bring Garrett back. Does that, so ultimately it is lifestyle, you know? I think we lost Garrett, but that's okay. Let's go to Bonnie Jones. Go ahead, Bonnie. Bonnie, go ahead. Oh, we're losing you, Bonnie. We don't hear you. You're going in and out, Bonnie. Oh, who's that? Prabhakar, who's that?
Not sure. I think it's.
I don't even hear who that is. Huh. Hold on, let me mute everyone.
And I also just
Bonnie, we're losing you. No one can hear.
Oh,
Bonnie, we couldn't hear you at all. Were you, I don't know if you're driving. Go ahead. Oops. Go ahead, Bonnie. No, we can't hear you, Bonnie. Mute her. Okay, Bonnie, maybe you can text us. Bernardo, go ahead. Bernardo?
Muy buenas tardes, Dr. Shiva. Gusto en saludarle.
Yes, what's your question, Bernardo?
How can I help CytoSolve to, I was reading the leukemia report that was sent to me and how the T cell one and everything acts upon the mutation cells, and I want to continue that and further and help further that research, also with the cannabis molecule of THC, sir. So
yeah, so I think we had I think we had a meeting with you, right? But yes, I think Prabhakar put together a proposal that's the next follow-up, Bernardo, that I would set up with Dr. Prabhakar.
Dr. Prabhakar, thank you. And
yep,
nice to talk to you again, too.
Thank you. Yep.
Dr. Shiva, it's Bonnie. Can you hear me?
Yeah, much better. Go ahead, Bonnie.
Okay, thank you. I wanted to say thank you for an extraordinary talk. I was at the Systems Health training in February, and I was very interested in learning more about CytoSolve. And as I learn more and more about it, it's more and more fascinating and interesting. And so I just want to say thank you for expanding on it today, and that the Systems Health course was absolutely amazing and very fundamental to a lot of the overall health interest and research and so forth that I enjoy. So thank you for that. And then number two, I wanted to ask you about, there's a gal here in Florida where I'm located. I think you know Linda Callahan. She works in psychiatric medicine and care. And so she has expressed some interest. She wasn't able to be here today, but she has expressed some interest in, you know, looking at how certain chemical pharmaceuticals can affect people. And so I just wanted to mention that and make sure she can contact you about that. Is that correct?
Yeah, so Bonnie, so in the area of psychiatric drugs, many years, so I live in a place called Belmont, Mass. Right behind my home is around 400 acres of conservation land, and on that conservation land is a McLean hospital. McLean is a Harvard hospital which does all this clinical work on giving people psychiatric drugs. Okay.
Yeah.
There were two Harvard professors, or husband and wife team, that I met many years ago, and they were like one of the biggest prescribers of these psychiatric drugs, and they actually saw them causing so many side effects and being ineffective that they left. And when I met them, this is 6-7 years ago, that was before I was heading out to California, they wanted
to really start doing work with CytoSolve on combinations of natural medicines. And they found a lot of value in things like rhodiola, for example, okay, among other things. So anyone in the psychiatric field can work with us. Again, this is why there's so many ideas, Bonnie, that we have to get you guys up to speed on what we can do. And you guys can become the philanthropists. You guys can go raise the money. You guys can be the research leads on this, right? So that's what she can do. If she's connected, there's, and then what's happening in the psychiatric field is people are just saying, oh, we're going to take these psychotropes. Let's do mushrooms. Let's do this. Let's do that. Right. But the problem with many of these herbal ingredients is there's no standardization. So people are into ayahuasca, they take it. And then a friend of mine, she said, oh, I'm going to do ayahuasca to stop smoking. All right. She went to do that. She became a chain smoker after that.
Oh, good God.
Yeah, literally. So you see these extremes that are occurring because these things have different effects on different people. So that's why we have the Systems Health Platform and CytoSolve to really figure out, because we want to be prudent, right? We have to do better than what Big Vitamin does and what Big Pharma does. And that's what we can do. But so what I would do, Bonnie, is give her the Calendly link and we can do a follow-up with her.
Okay, beautiful. That sounds great.
Yep.
Thank you, Dr. Shiva.
All right, we'll take last hand by Linda Scalia out of Alberta, Canada. Go ahead, Linda. How are you? Linda? Linda
Hi.
Scalia? Hi, Linda.
No, this is Laura. Dr. Shiva, this is Laura from Calgary.
Yes, Laura, how are you?
Good, thank you. I just want to go back to the MV25 and just ask you, was there a specific purpose for adding the vitamin C to the mix?
Yeah, so the vitamin C, it supports, it's also a bioflavonoid. If you go, let me give you an example. So many of these bioflavonoids work synergistically. So you can, for example, by the way, on a separate note, if you go just take vitamin C, it's not as good as taking vitamin C with bioflavonoids. So vitamin C helps accentuate the hesperidin, Laura. That's why.
Oh, okay. So it helps to metabolize the two.
Yeah, it makes it more bioavailable. Yep.
And just a quick question: did you say so we can attend the course there, like the whole 3 days online?
Yes, we made it online now. So the feedback we got from the first workshop we did was in February, was that some people wanted to make it online, other people wanted payment plans. So that's what we did. It was a lot of work figuring all this out, but now we've made it accessible to people because some people can't. I mean, we have to keep our lights on. So there's a certain set of things that we do. So we want to incent people to become warriors because then they learn to fight, to heal, and to think. So we gave a deep scholarship there. And then on top of it, we gave the payment plan. And then on top of it, we're making it so people can, let's say, just come to day one. And by the way, if you just come to day one, whatever you spend, you can then use it for day 2 and day 3. You say? So yes, Laura, we did that.
So
all right,
is the
everyone.
cost the same to attend online?
Same cost, yeah, same cost.
Okay.
Because the reason we did that is once you pay for day 1, in a year you can attend as many day 1s as you want. So if you decide to come in, you can reuse the pass.
Okay, thank you.
So, I mean, the difference in attending is it's more of a personal thing, but we also, the difference is we bring in lunch, and then on one of the days, we go out to dinner. So it's a little bit more team, and you're physically here in Cambridge. All right, everyone, in the interest of time, we've been here now for 2.5 hours. I'm sure we could all keep going. But again, the to-do items are, Prabhakar, you've gotten a number of people's phone numbers, right, Prabhakar? We have some follow-ups?
Yeah. Well, I have got one phone number so far. I haven't got any, but I've noted the names of people who are interested.
Mukibi. Okay, he didn't give you his number? Okay.
No, but I can get the email from Prakash.
Manju. Yep.
So to all of you, this is the CytoSolve Symposium. We'll be doing one next month. Next month, we're going to be talking about lung issues, lung health. Pollution globally is the #1 source of death right now, pollution. But the issue is what toxins affect it. So we've been doing a lot of research on all the different lung diseases. So someone, what is Ken saying? Chi-ST is disabled? I don't know what he's talking about. Ken, what are you talking about? Chi-ST? Ken? Oh. No, chat is not disabled, Ken. People can only chat to the hosts. It's disabled for each people, so they don't waste their time talking among each other, right, Prabhakar? So people can chat directly to you. Go to truthfreedomhealth.com. One way you support yourself and everything we're doing is become a Truth Freedom Health Warrior Scholar. You can also go to the International Center for Integrative Systems. Emily put out a chat. Donate to support this research. Third thing is become part of the change. It'll be new, it'll be exciting, it'll be a little scary, but you yourselves can be supporting science. The last piece is Systems Health, May 24th, 25th, 26th. We have our summit, which is part of being a Systems Health educator. It's an amazing opportunity for you to learn more. You can support your health as well as others. Prabhakar, is there anything else that you want me to share with people before we sign off for today?
I think that's it.
Okay. Prabhakar, I would download the chat so you get everyone, like Shanaz's email, all that. I think you got her.
Yeah.
All right, everyone. Thank you. Be well. Get involved. Get involved. Get involved. By the way, those of you who are in the United States know that I am running for president. We need to get signatures to get on the ballot. You can go to shivaforpresident.com. Support us. I don't think there's not any other presidential candidate who comes from my background as a skill sets that we have, and everyone deserves that. They don't just need someone who's quote unquote likable. They need people who are going to actually get work done. Thank you. Bye-bye.